So this is the third part of lecture number 3. Okay, and in this video lecture, first I'll talk about some definitions, like definition of inoculation, growth, and identification. Okay, you should have a very clear idea about these terms, these terminologies. You know, okay, because these terminologies are very important in the study of microbiology. Okay. Okay, so then after this, I'll talk about how we can observe microorganisms, or more specifically, how we can observe bacteria under microscope. We cannot see bacteria in our naked eyes. We have to use microscope. Then I'll talk about vaccination and finally chemotherapy. Okay, so first the terminology. What is inoculation? Okay, inoculation is introduction of organisms into a defined location. For example, when a sample is added to a sterile medium, it is inoculation of culture. When organisms or protein components are introduced into animal body for protective immunity, it is immunological inoculation. So what inoculation means? So for example, you have some bacteria, okay? You have E. coli, and you put that E. coli, or you have transferred some of the E. coli into a new media. Okay, that media can be solid media or liquid media. So you have some E. coli and you put that E. coli into that media. This is called inoculation. Okay, inoculation of culture. And another example is, say, suppose you have some antibody, okay, and you inject that antibody in a person's body, okay, or into an animal's body. This is also inoculation, but this will be called immunologic inoculation, because antibody, they are immunological agent. Okay. Okay, then growth. Okay, in general, what we understand about growth? Suppose if I say, what do you understand growth, if you mean human? So in case of human, growth means increase in size, right? Increase in length, increase in size. But in case of microbiology, growth doesn't means increase in size. Okay, in microbiology, growth means increase in the number of cells. Okay, so that means from one bacteria to two bacteria become four bacteria, four become eight. So this, okay, so in microbiology, growth means increase in the number of cells. Okay, okay, finally, identification. We know what is identity, right? Identify means to identify something. Okay, like that means, okay, this is this. Okay, so in microbiology, identifications means actually identify a microorganisms. In in what basis? Okay, what are the basis by which you identify a microorganism? So identification in microbiology means detecting the taxonomy. Taxonomy means nomenclature. Like, okay, the taxonomy, genetic makeup, that means genetic material, metabolic pattern, and virulence factor of an organism. Okay, so that means identification in microbiology means detecting the taxonomy. I mean, what is there? I mean, in what type of species there? In what genus it is? Genetic makeup. I mean, it's DNA sequence. Okay, what type of DNA it has? Metabolic pattern. Okay, metabolic pattern. I think I don't know whether you are familiar with this word, but metabolic pattern means, like, for example, every species actually they have different metabolism. Like we human, we have different metabolism than the like cow. Okay, so in also it's true for microorganism. Every microorganism, they have different type of metabolism. Okay, some may metabolite cellulose, some cannot metabolite cellulose, something like this. Okay, some may metabolize lactose, other cannot metabolize lactose. So every microorganisms, every bacteria, they have different metabolic pattern. And finally, virulence factor. Okay, so virulence factors means, like, say for example, virulence factors are the weapon. So what kind of weapon? Okay, say E. coli. Okay, E. coli is a bacteria. You know, there are many different E. coli, different types of E. coli, and all the E. coli, they don't cause disease. Some E. coli cause disease. So why or how some E. coli cause disease? So some E. coli, they have some weapons. By this, using these weapons, they can cause disease. So the E. coli who lack these weapons, who don't have these weapons, they cannot cause the disease. So these weapons by which organism cause disease are called virulence factors. Okay. So again, I will read the definition. So definition of identification. Okay, so identification in microbiology means detecting the taxonomy, genetic makeup, metabolic pattern, and valence factor of an organism. Okay, so for example, an unknown organism should be grown in culture, observed under the microscope, biochemically tested for metabolic pattern, and analyze the DNA, hybridization for genetic match, and the virulence factor should be checked by PCR to identify an organism. Okay, in this part, in this by this sentence, actually each wanted to describe how we can detect this. Okay, so for example, we want to know what kind of say you want to detect a bacteria. Okay, so you want to know what kind of bacteria it is. So what first you have to do? First you have to culture that bacteria. That means you have to grow that bacteria. Then you have to see it, observe it under microscope. So if you observe it under microscope, then you will know what shape it has. Okay, what type of bacteria it is. You will have some idea. Then you got the morphology. Okay, so then you want to know its metabolic pattern. So how you can know its metabolic pattern? By doing some biochemical test. Okay, there are actually several tests. By doing that test, we can get the metabolic pattern of that bacteria. Okay, then you want to know its genetic makeup. How we can do this? Okay, although it's written here like analyze DNA by hybridization, but basically nowadays we just can sequence them. You heard the name, right? I think nowadays you frequently heard this sentence that DNA sequencing, DNA sequencing. Okay, many people are sequencing the virus, the bacteria. So just by doing sequencing, we actually can get the total genetic makeup. Okay, and finally, you want to detect the virulence factor. So how we can detect the virulence factors? Okay, actually, there are different methods to detect the virulence factors. One very common method is by using PCR. So, right now you know that by using PCR we can detect pathogen. For example, this COVID-19. Okay, so we all know the COVID-19. So, and we also know the PCR. At least now we know that by doing PCR we can detect COVID-19. But this PCR techniques, it has several other applications. By using PCR we can also detect virulence factors. Okay. Okay, so anyway, please try to have a clear idea about this terminologies. Okay, now how we can observe the microorganisms, say bacteria. Okay, you want to observe bacteria. How you can do that? Bacteria are very small. We cannot see them by naked eyes. So you culture the bacteria and you got this one. Okay, like this, something like this. Okay, or something like this. These are bacteria, we know. But are you seeing a single bacteria there? No. They are cluster of bacteria. By naked eyes, we can see that there are some bacteria, but we have no idea how actually they look because these are cluster of bacteria. We don't know how a single bacteria looks. So how we can do that? We can observe them under microscope. So you just cannot observe the bacteria under microscope. Before, you observe them under microscope, you need to do some preliminary thing. Okay, you have to prepare the bacteria, then you can observe them under microscope. Okay, so there are actually different ways to do that, but the most common way has been described here. So let's see what is the most common way to prepare our bacteria to observe under microscope. So what we have to do, say you have this one. You cultured your bacteria and you got this one, some isolated colony of the bacteria. So what you have to do first, take a glass slide. We all know what glass slide is, right? So take a glass slide and take some, put 1 drop of water or normal saline here. Take the slide, put 1 drop of water and normal saline here. Then we have a special device which is called loop, inoculation loop. Okay. So after the varsity open, I'll show you how these things look. So okay, these are like needle like things. Okay, so you take your slide, you put 1 drop of water or normal saline here, then use this loop and take very tiny amount of bacteria from here. Okay, don't take too much. I mean, this one the colony is contain millions of bacteria. So just took tiny amount of it's very tiny amount of bacteria from here using this special device which is called loop. Okay, and then put that bacteria here in the sample. Then what you have to do? You have to spread it. Okay, you need to spread it uh, onto that slide. Okay, like this way. Just spread them. Right? Okay. So after spreading them, this is still like it's not dry. Okay, it's contain the liquid. So what you have to do? You have to air dry them. Air dry them just keep it in the air so the water will get evaporated. Okay. So now you have a dry slide. Okay, dry slide which has bacteria on it. Then what you have to do? You have to fix them. Just hit the slide this way, so your bacteria slide cells they will get attached to that glass slide. Okay, and then actually after this method we can do some staining. What staining means, like, okay, you did all these things and you just now you want to observe this one. So if you just take this slide and put it under microscope, actually you will not see anything because bacteria transparent. You will not see any color, anything there. It will be very difficult to see anything. So what we have to do, we fix the bacteria onto a glass slide. So now we have to color the bacteria. We know color, right? রং করা মানে আমরা সবাই জানি না, হ্যাঁ, কালার পেন্সিল দিয়ে আমরা রং করি। Okay, so actually there are some similar methods by which we can color our bacteria. Okay, and there is a very basic method which is called gram staining. Okay, and if we do gram staining, there are two major types of bacteria, gram negative and gram positive, and there is a staining coloring method by which we can color this bacteria in different color. Okay, but anyway, I will explain about gram negative and gram positive bacteria in a different lecture. Okay, so just now remember this. The bacteria, they're transplant. They're transparent actually. If you just put them under microscope, you will see nothing. So after processing your bacteria in this way, you have to do some coloring method here. You have to use some coloring techniques to color your bacteria. Then you can put them under microscope, and in the microscope you will observe something like this. Okay, so these are actually the real bacteria. Okay. And bacteria have different shape and morphology. The most common shape is either their round shape or this kind of shape. Okay, these shapes are called rod shape. Okay, this rod shape, these are called bacillus, and this round shape is called coccus. So coccus means round and bacilli means rod shape. Okay. Okay, so we learned how we can observe bacteria under microscope. In the next video, I'll talk about vaccination and then chemotherapy. Till then, be safe.